Neurons, or nerve cells, carry out the functions of the nervous system by conducting nerve impulses. They are highly specialized and amitotic. This means that if a neuron is destroyed, it cannot be replaced because neurons do not go through mitosis. There are some exceptions, though, such as neural stem cells and progenitor cells in certain regions of the brain, like the hippocampus, where neurogenesis, creation of new neurons, is possible. Here, we will focus on neuron basics. Neuron consists of three main parts. 1. The cell body, soma, which contains the nucleus and organelles. 2. Dendrites, which are branching extensions that receive signals from other neurons. And 3. The axon, a long projection that transmits signals away from the cell body to other neurons or muscles. The axon often ends in axon terminals, which release neurotransmitters to communicate with other cells. Additionally, many axons are protected by Schwann cells covered by a myelin sheath, which insulate the axon and speeds up signal transmission. The gaps in the myelin sheath, known as nodes of Ranvier, are crucial for the rapid propagation of action potentials. Now for some more details on each of the parts. The soma contains the nucleus, which holds the cell's genetic material, DNA, and controls the cell's activities, including growth and metabolism. The ribosomes inside soma is the site where most of the neuron's proteins are synthesized. These proteins are essential for the maintenance and function of the neuron. The soma also integrates incoming signals from the dendrites. If the combined signals are strong enough, they trigger an action potential that travels down the axon. Finally, the soma contains various organelles, such as mitochondria, which provide the energy needed for the neuron's activities, but also Golgi apparatus, ribosomes, endoplasmic reticulum. Dendrites receive chemical signals, neurotransmitters, released by the axon terminals of other neurons at synapses. We will explain synapses later. These signals are then converted into electrical impulses. The branching structure of dendrites increases the surface area of the neuron, allowing it to form more synaptic connections with other neurons. This enhances the neuron's ability to receive and process information. Dendrites integrate the incoming signals from multiple neurons. The combined electrical impulses are transmitted to the soma, where they are further processed. Dendrites can also change their shape and structure in response to learning and experience, a process known as synaptic plasticity. This adaptability is crucial for memory formation and learning. Let's move to axon. Axon hillock is the initial segment where the axon originates from the soma. The axon hillock is where the decision to generate an action potential, electrical impulse, is made. It integrates incoming signals from the dendrites and soma, and if the combined signal strength exceeds a certain threshold, an action potential is initiated. Further down the axon, we find Schwann cells, a type of glial cell found in the peripheral nervous system, PNS. Their primary function is to support and insulate nerve fibers by forming the myelin sheath around axons. Myelin sheath insulates axons, preventing electrical signals from dissipating as they travel along the nerve fiber. It also significantly increases the speed of electrical impulse transmission through a process called saltatory conduction, where the impulse jumps from one node of Ranvier to the next. Schwann cells provide structural support and protection to neurons, ensuring their proper functioning and survival. In the event of nerve damage, Schwann cells play a crucial role in the repair process. They help clear debris and guide the regrowth of axons, facilitating the recovery of nerve function. They also supply nutrients and remove waste products from neurons, contributing to the overall health of the nervous system. The nodes of Ranvier are small gaps in the myelin sheath along an axon where the axon membrane is exposed. The nodes contain a high density of voltage-gated sodium and potassium channels, which are essential for regenerating the action potential. Regenerating the action potential at the nodes of Ranvier can be compared to boosting a Wi-Fi signal with extra repeaters. This ensures that the signal remains strong and can travel quickly along the axon. Now, we mentioned that in saltatory conduction, the impulse jumps from one node of Ranvier to the next. Let's clarify this. The term saltatory conduction comes from the Latin word saltare, which means to jump or to leap. This term can be a bit misleading because the electrical signal doesn't literally jump in the physical sense. While the signal doesn't physically leap, The effect of the rapid transmission and regeneration at the nodes gives the impression of jumping. The jumping metaphor is used to describe the way the signal moves quickly from one node to the next, making the process much faster than continuous conduction in unmyelinated axons. Here, the sodium-potassium pump then works to restore the original ion concentration gradients by pumping sodium ions out and potassium ions back in, ensuring the neuron is ready for the next action potential. We will talk about sodium-potassium pump more later. And now, axon terminals. They convert the electrical signal traveling down the axon into a chemical signal that can cross the synaptic gap to the next neuron, muscle, or gland. Axon terminals contain synaptic vesicles filled with neurotransmitters. When an action potential reaches the axon terminal, it causes the membrane potential to change. This change in membrane potential opens voltage-gated calcium channels in the axon terminal membrane, allowing calcium ions to enter the terminal, which triggers synaptic vesicles that contain neurotransmitters to move towards and fuse with the presynaptic membrane. The fusion of the vesicles with the membrane releases the neurotransmitters into the synaptic cleft, i.e., the gap between the presynaptic and postsynaptic neurons, just another name for it. After the release of neurotransmitters, the calcium ions that entered the axon terminal need to be cleared to reset the neuron for the next action potential. This is done in several ways, by calcium pumps, which are specialized proteins that actively transport calcium ions, or by calcium buffering, where intercellular proteins bind to calcium to help regulate its concentration, or lastly, by sequestration into organelles to be used for their purposes. Yes, not just soma, but the axon terminal also contains several organelles, such as mitochondria, needed to provide the energy required for various processes, including the active transport of ions and the synthesis of neurotransmitters. Endoplasmic reticulum, involved in calcium storage and release, as well as the synthesis of proteins and lipids. And synaptic vesicles, which store neurotransmitters. So, we've explored the structure and function of neurons, focusing on key components such as the soma, dendrites, and axon. Each part plays a vital role in the complex process of neuronal communication. Now, let's just quickly explain the synapse. A synapse is the junction between two neurons, or between a neuron and another cell, such as a muscle or gland cell, where communication occurs. It consists of three main components. 1. Presynaptic terminal, the end of the axon of the transmitting or presynaptic neuron, which releases neurotransmitters from the small sacs called synaptic vesicles. 2. Synaptic cleft, the small gap between the presynaptic terminal and the postsynaptic membrane. 3. Postsynaptic membrane, the part of the receiving or postsynaptic neuron or cell that contains receptors, special proteins for the neurotransmitters. When an action potential reaches the presynaptic terminal, it triggers the release of neurotransmitters into the synaptic cleft. These neurotransmitters then bind to receptors on the postsynaptic membrane, allowing the signal to be transmitted to the next cell, and the process we just explained repeats in the next cell. Synaptic plasticity is the ability of synapses to strengthen or weaken over time, which is crucial for learning and memory. When a synapse is repeatedly stimulated at a high frequency, it leads to a sustained increase in synaptic strength, for example, by insertion of more receptors into the postsynaptic membrane. We call this long-term potentiation, or LTP. On the other hand, when a synapse is stimulated at a low frequency over a prolonged period, it results in a decrease in synaptic strength. This we call long-term depression, or LTD. Just like physical exercise is essential for our bodies, mental exercise is vital for our brains. Our body aims for efficiency. What is not used gets discarded. Lack of mental activity can lead to brain atrophy. Without regular mental stimulation, cognitive decline can begin as early as our 30s. If you enjoyed this, don't forget to like, share, and subscribe to stay tuned for more updates.